Hello everybody, my name is Bianca and I'm a researcher at the Institute for Terrestrial and Aquatic Wildlife Research in Germany. Today I'm very proud to tell you a bit more on our research on marine litter in German waters and its consequences for marine mammals. But first of all, let me give you a little overview on our institute. So our institute, the Institute for Terrestrial and Aquatic Wildlife Research, is based in Büsum, in the very northern part of Germany, located at the North Sea coastline. But we belong to the University of Veterinary Medicine, which is based in Hanover. We do have different interdisciplinary research and in different scientific fields, such as investigation on the health status, bioacoustics, biodiversity, the investigation on the habitat use of marine mammals, and the effects of anthropogenic interferences, just to name some of them. In Germany, we have 3 different federal states which are located either on the North Sea or the Baltic Sea coastline. Here we want to concentrate now on Schleswig-Holstein, in which also Büsum is located. As you can see, the federal state of Schleswig-Holstein do have a coastline both to the North Sea shore and the Baltic Sea shore, which is shown here in pink. It is very specific because we do have a so-called stranding network here, covering both the North Sea coastline and the Baltic Sea coastline. A stranding network means that carcasses which are washed ashore in both coastlines are transported to ITA for further investigations. We have to keep in mind that necropsies are the best approach to learn more about the health status of our marine mammals. But now let's have a closer look on the topic of marine litter. During my PhD study, I investigated the distribution and occurrence in German waters and its impacts on marine mammals. In order to learn more about the situation of marine litter pollution and its impacts, we evaluated all available datasets to capture the whole picture. This includes different datasets. One of them is the aerial survey dataset for floating marine debris, macrolitter findings in marine mammals in course of necropsies, and also investigations on feces sample also taken during regularly conducted necropsies. In German waters, we do have 3 marine mammal species which occur regularly in our waters. This is the harbor porpoise, the harbor seal, and the gray seal. We have to keep in mind that all 3 species are highly mobile and protected under European law, such as the Habitats Directive. So let's come to the first data set I was talking about. So since 2002, we do aerial surveys to investigate the abundance and distribution of harbor porpoises. In the course of that, we also collect information on floating marine litter. For that, we do use the line transect distance sampling with a constant altitude and speed during our surveys. We do our detections with the naked eye. Just to give you an idea on what we actually see while we are flying, we can see everything which is larger than a milk cartoon. We can distinguish between different materials, such as processed wood or plastics. We record the time and the GPS position, and we can categorize the objects, either if it's fishing-related debris or debris of households or industrial waste. For our evaluation, we used all data collected between 2002 and 2016. We wanted to evaluate the so-called average encounter rate, which means all items we've spotted per kilometer. We had the exact numbers to compare the occurrence of marine litter both in the North and Baltic Sea, and we wanted to visualize our findings by creating maps and distinguish between different distribution patterns. So what you can see here is a map with the 10 by 10 kilometer grid cells showing the average encounter rate of floating debris objects in German waters. Average encounter rate means that the number of items per distance covered are shown here. The darker the grid cell, the higher the number of items per kilometer. The map shows also the nature conservation sites. Here in the North Sea, it's the Dogger Bank, the Sylt Outer Reef, and the Borkum Reef Ground. In the Baltic Sea, it's the Fehmarnbelt, the Kader Trench, and the Odra Bank. Furthermore, we can see that the average encounter rate is higher in offshore areas compared to coastal areas, both in the North and the Baltic Sea, and also within the nature conservation sites, which builds a high risk for protected species. The second topic deals with marine litter occurrence in marine mammals, and therefore we checked our necropsy protocol since 1990 or for seals 1995. We checked the database for all litter items which were collected, either general or fishing related, and we wanted to know more about the microscopical and histopathological findings in the marine mammals. We also needed a base date for the number of animals necropsied on the GIT, the gastrointestinal tract, to know how many animals are actually impacted. So we were keen on the information on the items, but also the associated lesions. So what we found was we had 14 external findings and 17 internal findings for our marine mammals. In total, we found 37 items. 25% of all collected animals were actually dissected on the GIT, and it were more collected in the North Sea compared to the Baltic Sea. In Schleswig-Holstein, we had also more animals collected and also dissected compared to the other federal state, Mecklenburg-Western Pomerania, which can be explained by the occurring stranding network in Schleswig-Holstein. The occurrence itself showed that gray seals were mostly affected while harbor porpoises were least affected compared to all three species. We had higher entanglement and ingestion in the Baltic Sea, and it was predominantly fishing-related debris we found. Apart from that, plastic items were the most occurring marine litter items we found in our marine mammals. So you learned now about the marine litter occurrence in our marine mammals, which are regularly appearing in German waters. But sometimes we do have also visitors, such as the sperm whale. In 2016, we had a mass stranding of sperm whales within the North Sea area, 16 of them stranded on the German coastline. In total, there were 30 animals stranding in the North Sea area in different countries, 16 in Germany, 6 in the Netherlands, 6 in Great Britain, 1 in France and 1 in Denmark. In total, 22 GITs could be examined and 9 animals found being affected by marine litter. 5 of them were located in Germany, 2 in the Netherlands, 1 in Great Britain and 1 in France. So what we found here in the course of this necropsies is a variety of different marine litter items, as you can see here, such as netting material, coffee capsule, sweep wrapping, rope, basket, agricultural foil, strapping tape, a fish hook, and long lines. But the most astonishing item we found was a car part from an SUV. In total, we found in all animals, of the 30 animals, we found 322 debris items. 78% were fishing-related litter items, such as netting material or fish hooks. One animal had up to 24 kilograms of debris in its stomach. But we have to keep in mind that the ingestion was not the cause of the death. The animals died due to the stranding process. But nevertheless, the findings we had showed that there is a risk because internal lesions can happen due to hard parts such as the car part or block the GIT, which could happen over the long run if the animals were having these objects over a longer time in their stomachs. So let's come to the next and last topic, the microplastic research in marine mammals from German waters. Since 2014, we collect samples during necropsies of marine mammals. And we collect both the intestinal samples and stomach contents. We store our samples in glass jars to make sure that we don't have any secondary pollution. We do have intestinal samples, including feces of all three species, which are collected during regularly conducted necropsies, but also scat samples of seals. For the intestinal samples, we do have a good availability and access to carcasses due to our stranding network. And for the scat samples of seals, we do have a health monitoring, which allows for collecting samples on sandbanks, which is a very non-invasive method to collect feces samples during this health monitoring. We wanted to know if there's a correlation between the health impact and plastic. So what we do in the necropsies is we collect feces samples within the gastrointestinal tract to make sure that we keep them as clean as possible and avoid secondary pollution. For that, we use acrylic hoods to make sure that we're just working in the most clean environment we can produce. What we do now is we collect the intestinal sample and we put them in washing bags. So we're sewing them in a specific washing bag to make sure nothing gets lost. And what we do then is we wash our samples in a regular washing machine using enzymatic washing powder. So we then filter our sample on cellulose filters and stain them with Nile red to use them under the fluorescence microscopy. So what you can see here is a variety of different pictures of potential microplastic particles. You need to know that Nile red is staining microplastics, and the more intense the coloring is, the more likely it is to have microplastics underneath the fluorescence microscope. What you see on the bottom left is a fiber, which most likely is a polymer. So when we're done with our first inspection under the fluorescence microscopy, we're using the Raman spectroscopy to find out more about the polymer we're dealing with. In this case, we do have the so-called spectrogram, which is like a fingerprint for all polymers we know. So this is a very clear idea on what polymer we are dealing with. So to give you an idea on some data we get out of this evaluation, we did some evaluation of harbor porpoise feces samples. And in this case, we used specimens which were collected between 2014 and 2018. We had 14 specimens from the North Sea and 16 from the Baltic Sea. In the North Sea specimen, there were 123 microplastics found. And in the Baltic Sea animals, it was 278 microplastic particles. We can see that in the North Sea specimen, 86% of the animals were impacted by marine microplastics, while in the Baltic Sea it was 100%. So all animals showed microplastic burdens. Comparing both seas, we can see that the burden in the Baltic Sea was higher than in the North Sea, and we don't have a real difference between male and female specimen. So to give you an idea on the polymer types we found, we had a look on 34 out of 69 investigated particles which were in the specimen from the Baltic Sea. And as you can see here in the picture, most polymers we found were polyester, which belonged to lost fibers of washed clothes. But we also had polyethylene and polypropylene, which is mostly used for packaging material. And we found PEST, PE, PA, and PP, which is actually related to fishing gear. So to sum that up, we investigated harbor porpoises both from the North and Baltic Sea, and we can see that both are exposed to microplastics. There was a significant higher burden in the individuals from the Baltic Sea, and it also coincide with the investigations of demersal and pelagic fish from colleagues, which show that they are also burdened by microplastics. There was no difference between sexes and ages, and we couldn't see any accumulation over the lifespan. It shows that egestion is clearly determined. The study also showed us first evidence that a good nutritional status is likely joined with a high quantity of microplastic. Healthy individuals feed more continuously, and so they swallow more prey which might be burdened by microplastics. So there is a high rate of ingestion and egestion. And it's also very important to consider that a favorite prey or feeding strategy might play a role by the amount of microplastics and the burden of the marine mammals. So with that, I would like to end my presentation. I hope I could give you a good overview on our research. And maybe you do have some questions, and I'm happy to talk about this topic with you in detail. Thank you very much.